Okay, so this data is also about nutrient cycles. It has important information at the top and also within the experiment as well. First questions identify the control variables, then we need to explain why they're important. So three main control variables in this experiment. So you used a 500 metre squared area, they used 30 days, and they did it in springtime. Why they're important, you need to think about what they're measuring. So they're going to be measuring the area covered by this weed, the pennywort, after they were treated in different ways. So 500 metre squared area is important because if they had a different area used, then it would affect the total area covered at the end. So if you had wanted a cleared 50 metre squared, then there's likely would have less area covered at the end. 30 days is important because if you left it for longer, then it's more likely that the weed would have regrown more, so more area would be covered. And it's important that it was done at the same time of year, so they have similar light intensity, similar temperatures. Both can affect the rate of photosynthesis, which how quickly the weed grows. So it would affect the total area covered if it was done at different times of year, because it would have regrown back at different rates. The second question, why the fewer fish with rivers containing the floating pennywort? The important information comes from up here. This weed forms a thick layer that floats on top of the water. So very, very similar to your eutrophication answer, which you'd have if nutrients were leached into water. So the thick layer of the weed is going to block light entering the water, so there's going to be less photosynthesis by the pondweed, the plants living under the water. If these will then die if they can't carry out enough photosynthesis. As a result, they will then be decomposed. The saprobionts, the decomposers, will use up oxygen within the water, so the fish are unable to aerobically respire, so the fish will then die. Question 3 asks for a percentage decrease for R. So it said the original investigation, it had 225 metres squared, area was covered with pennywort. At the end of the investigation, you can see that 160 metres squared area was covered with the floating pennywort. So percentage decrease is the decrease divided by the original value times 100. So it's gone from 225 to 160, so that's the decrease minus the 2, divided by the original, so what it decreased from, so it decreased from 225 times 100, so we had a 28.9% decrease. They could have also asked an extra question, so which was the best treatment, just using this graph. So you'd be looking for the one that had the lowest area covered at the end. You can see in here that would be P, so this looks like it's the most effective, because less of the weed has grown back. But there's no standard deviation bars or standard error bars, so you can't see if there's any overlap between these values. No statistical test has been carried out, and you also don't know the area that was covered if it was the same in each of the rivers to start off with. So something like a percentage decrease in area covered might have been better. Resource B then looks at P and Q. So these were chemicals that were, and they do seem to indicate that they reduce the amount of this weed. But do the chemicals also affect the number of insect species living in the river? There are other side effects to these chemicals that mean you wouldn't want to use them. So the next question is, describe what the data shows. So does it show an impact on the insect species? So first of all, we can see for P, there's been a mean decrease. For Q, there's been a mean decrease in the number of insects, so that's bad. So it's showing that both do decrease, showing a mean decrease in the number of insects. But is it a significant decrease? So you can see in P, if we take the standard deviation of this value, because this is the higher mean, we've got 28.6. If we add the standard deviation onto this, the lower mean, we've got 30.2, so this is now bigger than that. So we do have an overlap, so there is not a significant difference in P, because the standard deviations overlap. Q, there is a significant decrease though, so it's showing that there's a significant fall in the number of insect species. So a significant fall in the number of insect species in Q. Notice it's better to say a significant fall or a significant decrease than a significant difference. The last question is saying that P has less of an effect on insect species than chemical Q.
So is this the case? So evaluates agree and disagree points. So if the agree point that P has less of an impact on the insect species will be that there is not a significant difference or not a significant fall, so no real fall, whereas in Q there is a significant decrease in the insect species. There's also lots of other things which means we can't make any definite conclusion. So we don't know the number of individuals. It doesn't tell us anywhere how many insect species we're looking at. We've just got a mean. So it might not be representative. Also, if you remember from the previous experiment, and it says again here, it's only been done for 30 days. There's no long-term, we don't know the long-term effects of these chemicals. It could be that after the treatment's been applied, that the insects pick up again. Again, it's only been done in springtime, so the results could be different in another season. And we've only compared rivers P and Q. That's only 2 rivers. That's not necessarily representative of all rivers. And we've only had one trial of chemical P and another trial of chemical Q. So not representative at all.